Hello friends, welcome to BMH Learning. In this video, I'm going to talk about gas chromatography. It's a process which separates components from the given sample with a gaseous mobile phase. It involves a sample being vaporized and injected onto the head of the chromatographic column. The sample is transported through the column by the flow of inert gaseous mobile phase. There are two main types of gas chromatography. First is a gas-solid chromatography. Here the mobile phase is a gas, while the stationary phase is a solid. It's used for the separation of low molecular gases such as air components, hydrogen sulfide, CS2, carbon dioxide, carbon monoxides and oxides of nitrogen. The second is a gas-liquid chromatography. Here the mobile phase is a gas, while the stationary phase is a liquid, which is retained on the surface by getting adsorbed or through chemical bonding. Now let's talk about the principle of separation. The principle of separation in gas chromatography is partition. The mixture of components to be separated is first converted to vapors, and then these vapors are mixed with gaseous mobile phase. The component having more solubility in stationary phase will travel slower and gets eluted later. On the other hand, the component having less solubility in stationary phase will travel faster and gets eluted out first. So we can say the components in this technique are separated according to their partition coefficient. Now, what is this partition coefficient? Partition coefficient is the ratio of solubility of a substance distributed between two immiscible liquids at a constant temperature. Now let's talk about the components of gas chromatography. First is the carrier gas. Second thing here is sample injection port. Third is the columns. Then we have detectors. There are many detectors available like flame ionization detector, thermal conductivity, electron capture, nitrogen phosphorus, flame photometric, and photoionization. Now let's have a brief touch with every component. The carrier gas must fulfill the following criterion. It should be chemically inert. The gas should be cheap and readily available. It must be of high quality and should not result in fire accidents. It should give best possible results. Apart from these, it should also be suitable for both the sample to be analyzed and the detector being used. The commonly used gases are hydrogen, helium, nitrogen and carbon dioxide. Now, the hydrogen has both low density and better thermal conductivity, but it can react with unsaturated compounds. Also, hydrogen is inflammable and has explosive nature. Talking about nitrogen, it's cheap but its sensitivity is less. So that's why helium gas is preferred. Then we have the sampling unit. Sampling unit, or we can say the injection port, is attached to the column head. As we know, the sample must be in vaporized state. So the injection port contains an oven which helps to maintain the required temperature above the boiling point of the sample. The gaseous samples can be introduced with the help of a gas-tight needle having capacity of 0.5 to 10 ml. If the sample is in the form of liquid, micro syringes with capacity of 0.1 to 100 microliter can be used. Next component is the column unit. Now the column comes with different shapes and sizes, which includes U-tube type or coiled helix type. They are generally made up of metals like copper, stainless steel, aluminium, glass and nylon. Now there are two types of columns. First is packed columns. In gas-liquid chromatography, packed columns are densely packed with solid support which is coated with liquid stationary phase. The solid support should be finely divided and inert, and that's mainly provided by diatomies. Talking about gas-solid chromatography, the columns are packed with adsorbents or porous polymers. Length of these columns is 1.5 to 10 meter, and internal diameter is 2 to 4 millimeter. Second type of columns are capillary columns. The length generally ranges from 10 to 100 meter, and regarding inner diameter, it's generally 0.1 to 0.5 millimeter. Next component is the detectors. Once the solute particles are eluted along with the carrier gas, they have to exit from the column and finally they enter in the detector. The function of detector is to produce electrical signals which are proportional to the concentration of the solute. The electrical signals thus formed are then amplified and recorded as peaks at various intervals on the chromatograph. An ideal detector must have the following parameters. It must be sensitive and should operate at high temperature. It should be stable and reproducible. It should give linear response. An ideal detector must have wide dynamic range. It should give fast response. It should be non-destructive and also should give uniform response to all analytes. The technique has many applications. It helps in the analysis of many drugs like antibiotics, antivirals, general anesthetics, sedatives or hypnotics. The method can be used for drug assay, and by doing this, purity of a compound can be checked. This is used in the determination of the levels of metabolites in body fluids. It's very helpful in the analysis of many food products. Environmental pollutants like formaldehyde, carbon monoxide, benzene, DDT can be analyzed with the help of this technique. This was all. Thanks for watching.